So it's a pleasure to be with the group to give this presentation. I'm going to try and go through just at high level some of the lessons we learned or lessons we relearned from studies of the mechanism of action and the PK/PD pharmacokinetic-pharmacodynamic relationships for antiviral medicines during the COVID-19 pandemic. I'm a professor of pharmacology at the University of Liverpool, and I'm also a director of the Centre of Excellence in Long-Acting Therapeutics. So I'll start with just a few disclosures. And I just start off by saying, what did we learn, or what did we relearn in the first 12 months of the pandemic? On the slide here, I'm showing a drug development pyramid. This is what I've been taught over the last 25 years of my career. We start in preclinical development, and providing we have plausibility, we move into phase 1 clinical trials, from phase 1 into phase 2, and from phase 2 into phase 3. So what did the first 12 months of the pandemic feel like? Well, it felt a lot more jumbled than that. We had a lot of phase 3 trials happening, particularly with repurposed medicines. We had preclinical trying to backfill gaps in knowledge. We had a little bit of phase 1, a little bit of phase 2, but the predominant effort was in phase 3 evaluation of repurposed agents. I would highlight that in the background of that, there was classical drug development pyramids occurring for some of the molecules which later proved to be successful. And just to highlight that, we know that drugs like medicines like nirmatrelvir, ritonavir, Paxlovid, like molnupiravir, like sotrovimab, emerged from those classical approaches to drug development through the phases. Other molecules which went straight into phase 3 with things like hydroxychloroquine, lopinavir, ritonavir, sofosbuvir, ivermectin, etc., etc., etc. So what did we relearn in the first 12 months? The mechanism of action must be demonstrated, it must be plausible, and it must be tested in RCTs. Secondly, in vitro activity is highly relevant, but it has to be considered in the context of pharmacokinetics. So we need to understand the PK/PD. We need to understand the exposure response. Thirdly, repurposed drugs can be highly valuable, but only when appropriately validated and tested. And fourthly, non-randomized clinical evidence is unreliable, and obviously badly conducted RCTs are too. So I'm just going to go through some of the things that came across my radar during the pandemic, which highlight these points. So starting with the mechanism of action, we all know what happened with chloroquine and hydroxychloroquine early in the pandemic. And on the left here, I'm showing the original February 2020 paper, which demonstrated EC50 values in the low micromolar range for chloroquine against SARS-CoV-2 in culture in viral cells. And just a quote from the paper here, chloroquine is known to block virus infection by increasing endosomal pH required for virus cell fusion. Now, hydroxychloroquine went straight into clinical trials, but a year later, January 2021, we started to learn more about the mechanism of action of the 4-aminoquinolines. And this particular paper, which was very interesting, demonstrated that the viral cell line had an important mechanism which was absent, and that was via the TMPRSS2 mediated activation. And so there were two entry mechanisms for the virus, one via endosomal route and another which involved this TMPRSS2 mediated activation. And in cell systems and in vivo, where the TMPRSS2 mechanism was at play, the 4-aminoquinolines had much less activity. So phase 3 evaluation of the 4-aminoquinolines was underway prior to the emergence of that prerequisite knowledge about the mechanism of action. Secondly, in vitro activity is highly relevant, needs to be considered in the context of the pharmacokinetics. Now, this is a paper an analysis we undertook very early in the pandemic. So in the first 2 or 3 months of the pandemic, we assessed the in vitro activity available for all of the repurposed molecules for which in vitro activity was available. And we benchmarked those concentrations needed for antiviral activity against the known pharmacokinetics for those drugs. And you can see just highlighted on the side here, these drugs, I think there were around 60 molecules with reported activity at that time during the pandemic. And this is the Cmax, so the maximum concentrations in the bloodstream with these drugs versus the in vitro activity that had been reported. And you can see the overwhelming majority of molecules fell in a region where even at maximum concentrations demonstrated for antiviral activity weren't achieved at the approved doses of these drugs. And just to highlight, remdesivir, the measurements we had for remdesivir at the time were at or above, the concentrations were at or above antiviral concentrations, but a whole list of the other repurposed molecules, including hydroxychloroquine, were orders of logs lower than the antiviral concentrations. So sufficient data existed to conclude that hydroxychloroquine, lopinavir, and other repurposed drugs would not achieve antiviral concentration in the plasma of patients prior to the initiation of the clinical trials. It just wasn't harnessed in that way in order to inform candidate selection. It's also important to highlight that while initially studied for antiviral properties, proponents of drugs like hydroxychloroquine and ivermectin continue to argue in favor of secondary mechanisms of action, even when it was known that antiviral concentrations weren't achieved. Now, these secondary mechanisms of actions are usually things like disease-modifying mechanisms, immunomodulation, for example, which were plausible but unproven. There are other examples as well. Hydroxychloroquine was studied at a range of different doses, and this GIF here is flicking through all of the pharmacokinetic profiles for all of the different doses which are studied in various RCTs for hydroxychloroquine, and none of those regimens achieved concentrations which exceeded the in vitro activity measurements. There's another case here with the ProTide technology. So there's several drugs which are approved which capitalize on the ProTide technology. And these drugs are drugs like tenofovir alafenamide for HIV, sofosbuvir for HCV, but also includes remdesivir, which we're all aware of in the case of SARS-CoV-2. Now, the ProTide technology is distinct from other prodrug technologies because the objective is to deliver intact prodrug into the systemic compartment of the patient. And the active intracellular moiety is the same, but the ProTide intracellular accumulation and residence time is higher, which gives you an apparent more potent measurement in terms of the EC50 and EC90. And you can see that with approved drugs, tenofovir alafenamide against HIV, the plasma concentration exceeded the EC90, and the high intracellular concentration drives the efficacy across the dosing interval, despite the fact that the plasma concentration drops early. Similar with sofosbuvir and HCV. Similar with remdesivir, the remdesivir exceeds the EC90, and then the intracellular concentration drives the efficacy across the dosing interval. In the case of sofosbuvir in SARS-CoV-2, though, many argued for sofosbuvir in SARS-CoV-2, but the plasma concentration never achieved the EC90 target. So sufficient data existed to conclude that these drugs would not achieve antiviral concentration in plasma of patients prior to the initiation of the trials. The third point I wanted to make was repurposed drugs can be highly valuable, but only when appropriately validated and tested. And we know this because remdesivir was originally developed to treat hepatitis C virus infection and was also studied for Ebola and Marburg virus infections before being repurposed for SARS-CoV-2. Similarly, molnupiravir was designed as an orally administered influenza treatment and repurposed early in development as an antiviral for SARS-CoV-2. Nirmatrelvir was developed as an analog of an intravenous prodrug, which was lufotrelvir, which was originally developed for the original SARS-CoV and subsequently repurposed for SARS-CoV-2. And although development of sotrovimab only began in December 2019, sotrovimab was identified from the blood of a patient infected with SARS-CoV and not SARS-CoV-2. So repurposed drugs can be highly valuable, but they need to be validated properly. The fourth point I wanted to make was non-randomized clinical evidence is unreliable, and so are badly conducted RCTs. And I'll just highlight a comparison paper that was published by Andy Hill a couple of years ago. And I've just shown an excerpt from the table here for just ivermectin and hydroxychloroquine. And when you look at the observational study, the real-world data for these molecules, this suggested mortality benefits of 40% lower risk or a 49% lower risk for ivermectin and hydroxychloroquine. When the RCTs were conducted, there was no significant difference between these drugs and the placebo. Now, the reason for this obviously is that no statistical approach can account for unmeasured or unknown prognostic factors between groups, also known as residual confounding. The only way to deal with residual confounding is in randomization. So if RCT evidence is not available, it's impossible to determine whether the adjusted estimates are accurate or misleading, because you can't account for these unknown confounders when you do an analysis in an observational study. So I'm going to go through some of the molecules which I've mentioned already. Sotrovimab clearly had extremely supportive preclinical evidence for original variants of the SARS-CoV-2 virus. It's an Fc-engineered IgG. It's neutralizing. It's a human monoclonal antibody. It binds up to an epitope in the spike protein. And the antiviral activity in Syrian golden hamsters was clear early in the pandemic against the Wuhan variant. It was demonstrated at 5 mg per kg intraperitoneally. And neutralization of the Wuhan SARS-CoV-2 was achieved in VOE6 cells with an EC90 of 0.19 micrograms per mil. But considerable reduction in neutralization was widely documented for all variants since the BA2 Omicron, emergence of BA2 Omicron. Now, sotrovimab serum concentration in COMET ICE at the approved single 500 mg IV infusion provided mean Cmax comfortably above the EC90 value of 0.19 micrograms per mil. But unlike small molecule drugs, monoclonal antibodies have an extremely poor penetration into the target tissues. Now, there's been quite a lot of debate about whether sotrovimab is still active now against newer variants of SARS-CoV-2. And several recent preclinical studies showed virological efficacy of sotrovimab against the BQ.1.1 variant. But these were conducted in experimental designs reflective of prophylaxis for in hamsters and non-human primates. It's important to recognize that the bar for efficacy in prophylaxis is lower than the bar for efficacy in treatment, and there's several reasons for this. Viral inoculation occurs at maximum antibody concentrations when you conduct a prophylaxis design. So you're introducing the virus into the highest concentrations of the medicine. Efficacy only needs to suppress the upward sweep of replication in a prophylaxis design. Whereas in a treatment design where you introduce the drug after replication is established, you need to reverse that and you need to bring the viral load down. So we conducted a comparative assessment between treatment and prevention between Delta and BQ1.1 in immunocompromised versus healthy hamsters. And I'm just showing the data here. This is a lung viral RNA at the top for the Delta variant study, and at the bottom for the BQ1.1 variant study. And I'll just highlight that the sotrovimab, irrespective of whether it was prophylaxis or treatment, had a statistically significant effect on the viral load for the Delta variant in healthy animals and in immunocompromised animals. Whereas in the BQ1.1, with this reduced neutralization, there was an impact in the prophylaxis model for sotrovimab, but no statistical impact on the viral load in the treatment model, i.e., when the sotrovimab was introduced after infection had been established. So no virological efficacy against BQ1.1 in treatment of immunocompromised hamsters. So what do we understand about the preclinical PK/PD evidence for sotrovimab? Well, direct neutralization of the virus is the primary mechanism of action. In vitro neutralization measurements are used to define target exposures for clinical development. And based on publicly available information from the EMA, we're able to understand the target, the PK/PD assumptions, and how the target exposure of sotrovimab was calculated. Now, the calculation for the serum target included three factors. It included the in vitro neutralization activity using EC90, not EC50, as is convention when determining an antiviral target. It included an arbitrary tissue penetration to account for the low penetration into lung, and it included a margin of error to account for the variability in measurements and uncertainty regarding the tissue penetration. Now, importantly, up to and including BA1 Omicron, which had a 7-fold reduction in neutralization, all the variants that preceded that fell within that margin, such that the target concentration was still appropriate for those variants. But when BA2 Omicron emerged, the serum neutralization dropped between 20 and 50-fold, such that if the same calculation is employed using in vitro neutralization for BA2 or subsequent Omicron variants, then sotrovimab would be deemed not to reach the serum target. Now, this is important because intravenous sotrovimab is dose linear, it's dose proportional. So as you increase the dose, you increase the exposure linearly and proportionally. And therefore, a recommendation in favor of continued efficacy of sotrovimab against newer variants is quantitatively the same as a recommendation in favor of a reduction in dose from 500 to 25 milligrams for the Delta variant. Despite this, proponents for continued use of sotrovimab have argued for a secondary mechanism of action in the form of effector functions, again, which is plausible but unproven in RCTs. So when you look at the clinical PK/PD evidence for sotrovimab, this was an accelerated development program. So a lot of the early PK/PD studies weren't conducted because of the urgent public health need. However, there was one RCT, Comet Tail, which studied 250 milligrams IM, 500 milligrams IM, and 500 milligrams IV doses. And the Independent Safety Monitoring Committee for that trial terminated the 250 milligram IM arm due to a higher rate of hospitalization than either the 500 milligram IM or the 500 milligram IV dosing. So you can take the sotrovimab neutralization concentration, the plasma concentration, and divide it by the EC90 value and plot the data accordingly. And when you do this, you can see the red line here is the 250 IM delta, and that dictates the serum titer at which the hospitalization rate was higher, the efficacy was suboptimal in the Comet-Tail trial. And when you do the calculation for BA2 Omicron with the EC90 for the BA2 Omicron, you can see that the serum neutralization titer falls below that titer, which had already failed in the Comet-Tail trial. So based upon this analysis, policymakers with PK/PD expertise like the FDA, like the WHO, concluded that a 20-fold or greater reduction in neutralization would result in titers lower than the suboptimal efficacy, whereas other policymakers, based on observational data, made a recommendation to continue to use it. I would just highlight a paper that emerged today, actually, where in hematopoietic stem cell transplant recipients, it's been reported that the plasma concentration of sotrovimab are even lower than those in non-transplant patients, so even lower away from the serum neutralization titer. Now, there was non-randomized clinical evidence, observational data, which supported continued use of sotrovimab. I'm showing two studies here from Open Safely in the UK. The first one, where the main analysis was conducted during BA1 predominance, and not helpful in informing us of whether BA efficacy was maintained against BA2. Although there was an exploratory analysis during BA2, there was no therapeutic equipoise, so it's very difficult to make interpretations of the BA2 data in that paper. And then a second analysis, also from Open Safely platform, where the hazard ratios for sotrovimab were statistically significant for BA1, but not for BA2. Despite this, sotrovimab is still recommended for use in the UK. If we look at other small molecule antiviral medicines, nirmatrelvir, for example, we don't have the same issue. From the NIH NCAT database, you can see that the EC90 for Omicron variants cluster around 1, if anything, more potent or equivalent to against the Wuhan variant. Contrast that to sotrovimab, where there's a reduction in neutralization. And other data showing that the EC90s are very similar across the newer variants for nirmatrelvir. Similarly, for molnupiravir, consistently activity across newer variants of the virus. So major differences across the variants have occurred in the spike protein and not the protease of the polymerase, which are targeted by molnupiravir and nirmatrelvir. So activity of the small molecules has been more stable than that of the monoclonal antibodies. I did want to just highlight antiviral combinations. They didn't happen in the pandemic, but successful treatments for other viruses, HIV, HCV, involve the use of potent drug combinations. We conducted an analysis with colleagues at Drugs for Neglected Diseases Initiative of nirmatrelvir and molnupiravir, individually or in combination against BA1 Omicron in the K18 humanized mouse model. And you can see here that in the untreated animals or control animals, you get a high viral load. And you can see molnupiravir alone group here and the nirmatrelvir alone group here, both statistically significant differences in viral load. And then when you combine nirmatrelvir with molnupiravir, you get an even lower viral load. And if you look at viral titers by infectious virus, PFU, It was only the combination group where no replicative virus was detected in any of the animals. So combinations offer potential for higher efficacy, higher resilience against emergent variants if one drug gets compromised and not the other, as well as a mitigation of the resistance risk. So just in terms of summary and conclusions, the principles of antiviral pharmacology were borne out in the accelerated development of antivirals in SARS-CoV-2. It shouldn't be contentious that an antiviral intervention needs to reach antiviral concentration at its target site in order to exert antiviral efficacy, although it is in some cases. Small molecule drugs have proven resilient to changes in the virus over time, and all monoclonal antibodies have been compromised to some degree. And I'm just summarizing in the table here outcomes from PK/PD assessments, preclinical efficacy, RCT data, and observational data. And in the case of molnupiravir and nirmatrelvir, all of this data supports continued efficacy against new variants. If you look at sotrovimab, however, the PK/PD assessment does not support efficacy. The preclinical efficacy does not support efficacy in treatment, at least, although there is evidence that it may work in prophylaxis, short-term prophylaxis. If you look at the RCT evidence, there aren't any RCT evidence. The pivotal data are now indirect because it's a different virus with a different neutralization activity. And the observational data are mixed and variable outcomes. So continued efficacy of sotrovimab remains highly uncertain, but a definitive outcome is critically needed. I don't think it's going to be forthcoming. But I would pose the question, should we be using medicines with unknown efficacy? I think everybody can answer that for themselves. But I'll just leave you with the thought that, was the dose of sotrovimab really greater than or equal to 20-fold higher than it needed to be for pre-Omicron variants? And if so, we really do need to understand this. We need to understand the PK/PD so that we can use that information to improve equity and accessibility of other mAbs for infectious diseases in low and middle income countries.